Um, so this is our campus. On the left side, you can see the Antwerp University Hospital. And on the right side, you can see the building where we're at the fourth floor, the tumoroid screening lab. So you can see we're very close together. So it's very convenient for us to get samples from patients, biopsy resection fragments, and then process them in our lab to form patient-derived organoids or tumoroids. So just a small introduction about these tumoroids. So they're really 3D microtumors generated directly from patient samples. They've been shown to maintain the mutational landscape and also the clonal heterogeneity found in the tumor of the patient. So different subclones of cancer cells that could eventually lead to therapy resistance and so on. A very promising field is, of course, the clinical applications where you can predict therapy response on these organoids. So you have organoids, you can screen maybe 10, 20 compounds, see what works well, and then give this to the patient. And this concept is called functional precision medicine. But also, of course, for preclinical applications, these are very informative and very representative in vitro models. In my opinion, much better than the cancer cell lines that we have been using for a long time, because now we can really generate new biobanks of patient-derived organoids. We can generate our own biobanks. We can select, for example, from resistant patients we want to make organoids, or sensitive patients, compare this, and again, find new therapies and study the cancer biology. On the right, you can see, of course, the Clinostar that you all know, which is a recent addition to the automation family or the robotic family we have in our lab. And the second one is an OT2 pipetting robot that we use to dispense the organoid solutions in the 384 well plates, because of course it's very difficult to do this manually. We of course then also have our own in-house developed image analysis software, where we really aim to automate the entire process of image analysis, but also downstream data analysis and data visualization. The fourth equipment we have is the digital drug dispenser. So it's really developed with HP, where you print small drops, like small ink droplets, but now small drops of liquid containing your drug of interest, and you can treat an entire 384 well plate in a matter of seconds or minutes, depending on the number of compounds you want to test. And the last system we have is the Spark Cyto system, so a live cell imaging system that can capture high quality, bright field infrared images from our organoids. So this is a little bit the pipeline we have now. We harvest 3D organoids, pipette them, see the organoids, treat the organoids, image the organoids, analyze the organoids with our software, and correlate this back to the clinical response of the patient. So this is the conventional way for us to grow organoids. We grow them in these domes of extracellular matrix, like Cultrix from Maastricht. But you can already clearly see that there's an oxygen and nutrient gradient, because the organoids on the sides of the drop, they grow much larger, and the ones in the middle are much smaller. If we compare it into the Clinostar, we can see a drastic difference between the organoids. So both organoids were grown for, in this case, I think 13 days. And we didn't replenish the medium because it was just tests to see what would happen if we don't do it. But even after 13 days without refreshing the medium, the organoids looked healthy. They really grew large. Seems to be a very good way to accelerate the expansion of organoids for drug screening, which is of course very important when we want to apply this in the future for predicting therapy response in patients. The faster we can get the results to the oncologist, the better this will be for the patient.